I would like to introduce you to Christina Warinner, who is a pioneer in the study of ancient human microbiota. She earned her Ph.D. from Harvard University and received her postdoctoral training at the University of Zurich and the University of Oklahoma. She's an assistant professor of anthropology at Harvard University and a professor of biological science at Friedrich Schiller University in Jena. Since 2016, she holds a group leader position at the Max Planck Institute for the Science of Human History. She has published 2 books and many journal articles in highly distinguished journals such as Nature Genetics, PNAS, Current Biology, and Nature Communications. Professor Warinner has received also many awards and grants for her work. I cannot list all of them. This will take us a while. And she has also supervised several of our PhD students. Some of them you already had the pleasure to meet within the classes. So Sandra, James, Maxime, and Megan, who you will meet next week, are all PhD students of her. And in addition to her research, she's actively engaged in public outreach. I can highly recommend her TED Talk. And she also created Adventures in Archaeological Science, a coloring book, which is available in 30 languages, which is totally amazing for me as a linguist. I think it's really important, and some of them are indigenous and underrepresented. And yeah, so her TED Talk was about tracing ancient diseases using plague. And it's a pleasure to have you here, Christina. And I would, just for the students, we will have a 1 hour or so lecture. And then you also have the chance to ask a question to Christina. And so, but without further ado, I don't want to take too much of your time. The floor is yours, and please give it away.
Thank you so much for such a kind and wonderful introduction. I'm really happy to be here with you today. I'm so thankful that the organizer asked me to speak. Today I'm going to give a talk. It's different than the talks I usually give. Today, what I really wanted to focus on for students around the world is thinking about how do we work together? How do we actually create this knowledge of the past, of our shared human past? You know, how do we see together? And how do we bridge different disciplines, which is in many ways the spirit of this whole summer course? You know, how do we take genetics? How do we take stable isotopes? How do we take linguistics? And how do we put this all together and using all this diverse information, recreate the story of our past? So that's what I'm going to talk about a little bit today. I'm going to give a few examples of that. First, though, I just want to acknowledge how happy I am that we're all here and how difficult the last 18 months have been. I think it's really important to acknowledge that we're not in a normal research time. And I know many of you have had your research plans disrupted or slowed down or changed. 2020 was a pretty disruptive period. I can speak from my own experience here. For example, I was at Harvard teaching when we got a sudden announcement that all of our classes were going to move online with 4 days notice and that all of our students were being sent home. And this was totally shocking and really unprecedented since World War I at Harvard. I know that a lot of groups around the world responded really quickly. This is a picture of Luva Dahlin, who is a researcher in Stockholm, posted this on Twitter, that they were donating all of their equipment in the efforts to fight COVID-19 because of all the worldwide shortages. You know, we as archaeological sciences actually had stockpiled a lot of the PPE that different groups around the world needed. And our lab did the same thing. We packed up a lot of the materials that we had and sent them to local hospitals. But it was really disruptive. Labs shut down, everything stopped. No one quite knew what was going to happen. 2 of my students who were in Mongolia at the time doing fieldwork were unable to return, and their 6-week fieldwork turned into 6 months of fieldwork. So it was enormously disruptive this last year. And of course, for those of you who have been to ISBA or want to go to ISBA in the future, it's one of our largest conferences really on on archaeological science, biomolecular archaeology, was then all of a sudden canceled and postponed a whole year. So 2020 was just this year of change, surprise. It was a difficult year. And I know for those of you who had already started your graduate work, this might be kind of how you felt during that entire period. I really love this image because I felt like it really captured this period of us trying to figure out how the heck do we keep moving? How do we advance our projects? How do we finish things all at the same time while dealing with this, unfolding global pandemic? But I'm really happy that we're now in 2021, and I do feel like we finally see the light at the end of the tunnel. And so I think that events like this, like the summer school that we have right now, are so important to bring us all together after this year and a half of complete disruption, as we start to think about how do we continue our work on our important work on learning about the ancient past. How do we take lessons from what we've just been through? In many ways, there's parallels to what ancient people went through with different pandemics through history. But how do we also take these new electronic and digital forms of communication that we've learned about and maybe turn them into new ways of doing research? What can we take away from this? And I know that this week, a lot of what has focused on is how do we actually do the work of putting together the past? So how do we take information, samples, laboratory work, computational analyses? How do we do those? How do we make that happen? And then how do we turn that information that we gather into complete stories of the past? How do we reimagine and reconstruct these lost narratives, these lost or forgotten pasts? How do we put it all together? So I was asked to give the keynote today, and you might be wondering, well, who am I? And why do I care so much about this topic? This is something that I'm really passionate about. And in part, it has to do with my own background and how I came to be in this field that I am just thrilled to be a part of, because I think it's one of the most exciting and dynamic and important fields of science. And I'm really happy to be part of it. But what's my background and kind of how did I get here? So I actually I'm American. I'm from the United States, and I'm from Kansas, which is most people have only know of it through The Wizard of Oz. I went to school there. I went to the University of Kansas. But while I was there, I initially studied chemistry, but eventually took courses in archaeology and just was absolutely enthralled. I thought this was the most amazing thing I've ever learned about. And I very much wanted to be part of combining the sciences with archaeology to tell this past. I worked at a number of different Maya sites, and actually I did work in linguistics. I worked with Nikolai Gruba at the University of Bonn for a year, learning Maya epigraphy, and actually wrote a senior thesis on combining archaeology and Maya hieroglyphic and epigraphy to try to understand and reconstruct the lineages, the dynastic lineages of the ancient Maya. I then later went to Harvard as a PhD student, where I became even more involved in the archaeological sciences. I did a lot of work on mass spectrometry, in particular isotopic work, and also some work on ancient DNA. And I did a lot of fieldwork excavation as well in Mexico, which I'll talk about a little bit more in a minute. And then later I did a postdoc at the University of Zurich, and this was an incredible experience. I worked at the medical school there and was able to get more advanced training in proteomics, but also this brand new thing that came online while I was doing my postdoc, which was next generation sequencing for ancient DNA, which has truly, truly revolutionized our field. And I was really fortunate. I got involved with TED at that point and gave a TED talk and really started to develop my interests. Later, I went to the University of Oklahoma, first as a postdoc, later as a faculty member, and I worked at a large genomics center there. And built up their very first ancient DNA lab, which was a really difficult, but a really exciting opportunity as well, to build that really from the ground up. And we did, while I was in Oklahoma, a lot of research, but one of the most important for me personally was we worked a lot with Native American tribes based in Oklahoma, including the Cheyenne Arapaho tribes, to try to do collaborative, what would it mean to do collaborative, mutually beneficial research that was co-developed by different groups. During that time, I did a lot of work with the National Institutes of Health. I partnered with Illumina and TED and PBS, Nova to make documentaries. And so this public outreach aspect, communicating what we do in the labs with the broader world and why it matters has always been very important to me. especially because in a lot of archaeological science, so much of our work is done behind closed doors in clean rooms and other environments where the broader public doesn't get to see it. And yet the work we do is so exciting. So I'm always interested in ways that we can make this more transparent and really bring our research out of the lab and into the hands of people. And at that same time, I did a lot of traveling and giving guest lectures around the world, trying to get students excited about this new research and show them what the new possibilities were. And so here you see, this is 2 talks I gave, one in Mexico and one in China, talking about the intersection of archaeology and the sciences. And I had a really great opportunity. Actually, I was invited to go to the White House under the Obama administration to advise the government on how to look at the future of microbiome research, which is where a lot of my research is. And this is me standing outside the Eisenhower building, which is next to the White House, for this really amazing opportunity. And you'll see I am 36 weeks pregnant here. So I got to go fit it in right at the last moment, but it was a really great and exciting opportunity. And after that, I came to the Max Planck Institute, which was super exciting. I got recruited by Johannes Krause, who you see here, and I'm so excited to be a group leader here. I've been a group leader since 2016, and it's really an amazing experience putting so many incredibly talented people all together and all collaborating and working together on this kind of joint project of reconstructing the ancient past. And I built up a fantastic and amazing team of PhD students and postdocs who've just been incredible. And so a lot of the work that I'm going to talk about later and reference is actually the work that they have done. And so this is just amazing to be able to work in such an amazing team. And then while I was there, I also worked a lot with the ERC and the Deutsche Forschungsgemeinschaft, and did a lot of reviewing, for example, of Marie Curie applications for PhD students and postdocs. So sort of seeing the field at all these different levels. And so this has sort of informed a lot of what I'm going to talk about, what I think is important in the field and to think about. And then the last couple of years, our department has moved to the Max Planck Institute for Evolutionary Anthropology in Leipzig, and my group has moved too. And I've also taken a position as a faculty member at Harvard, where I also have students. So to me, this is just the opportunity, the ability to work with all these different incredibly talented young people and senior scholars all on this joint project is really amazing. And I think this collaborative spirit that characterizes archaeological science is one of the most exciting aspects of our field. In terms of research, a lot of the research I do is actually on kind of 4 main areas. I look at the evolution and ecology of the microbiome, the dairying and adaptive evolution in prehistory. I'm really interested in the prehistory of dairying and food technology. I'm interested in heirloom microbes and these sort of ancient microbes that humans have interacted with over a long time and how we've changed them. And then also human migration and adaptation. You know, how do people move around the world in prehistory, and how did that intersect with the spread of food technologies, the spread of ideas, and other factors? How could we understand how the present world came to be by reconstructing these deep histories? And as I said before, though, the greatest joy for me out of all of this is really the teams working with different people to solve these big questions. You know, how do you take on a major question about the past, and from just traces that remain, how do you reconstruct these stories? What is also exciting about archaeological science is the fact that we aren't just doing one thing. I mean, it's the intersection of material culture, excavation, linguistics, laboratory work. Everything comes together through these partnerships that cross-cut, you know, not only the sciences, but also the humanities, social sciences, natural sciences. We're one of the most collaborative and cross-disciplinary kind of fields of study. And of course, also the opportunity to train new people in the field, to have them come and learn new skills is so critical and important to think about the future of our field. So when people think about archaeology, or when they think about the past, they often think about images like this. This is a Hirixura from Mongolia, from northern Mongolia. It's a burial mound. And most people think of these kind of monumental scale architectures, these very visible, conspicuous signs of archaeology on the landscape. But for those of us in archaeological science, although this is a major part of archaeology, We also work at a different scale and with different types of materials, including things that may not seem terribly obvious or very small. A lot of our work focuses on the biochemical, the molecular, the things at a very, very small scale, like this tooth you see here, which also has dental calculus on a kind of calcified dental plaque. We can get so much information, not only from the large scale archaeology, but also from this very small molecular scale archaeology. And I often refer to this as the archaeology of the invisible, the archaeology of all the things that are not immediately obvious to your eye when you're looking at something, everything that needs to be perceived and detected using alternative methods. But where do we get this? How do we find, this? How do we retrieve this information, this archaeology of the invisible, from an archaeological site like this one here, which you see from Tapascaloola, Mexico? Well, I'm going to go over a few, and I think you've covered some of these already in the course. I mean, there's a few main sources that we go to recover this information, this molecular or biochemical level scale information about the past. Many of you know about the temporal bone of the cranium, and the petrous portion in particular, which we know is one of the densest part of the body, and it's an amazing source of ancient DNA. So if you want to study the genome of an ancient individual, or that's human or animal, that's going to be one of your best sources of DNA for reconstructing that genome. But there's lots of other things you can do with bones, for example. So here's a faunal collection. You can use bones for radiocarbon dating. You can look at stable isotope analysis to reconstruct aspects of diet or migration. There's just so many things you can do with these remains. I also work a lot with teeth. I think teeth are one of the most interesting parts of our body. You can learn so much from them. So for example, here, this is just a cross section of a tooth, and you can see its different components. It has many different parts, and you can get different information from it. So for example, here you see dental calculus, which I mentioned before, this calcified dental plaque. And here you can recover the oral microbiome, but also bits of food and other things that become entrapped during life. You see here the pulp chamber. You can see that it's fairly decomposed. You see that discoloration. But this is also really important because within here, this was full of blood during life. You can extract DNA from here also to reconstruct the host's genome from this individual. But also if they died of any kind of bloodborne infection, you can detect pathogen DNA that has decomposed and is now smeared along the interior of the pulp chamber. And so this is one of our best sources of DNA for reconstructing ancient disease. You can also look at the decomposition microbiome. So all this discoloration that you see here at the bottom of the tooth, that's caused by soil microbes that are slowly degrading this tooth, and you can magnify them and see their action today. We can learn a great deal of information from this. So as I mentioned before, looking at the oral microbiome, one example is a study recently published by my student James and also postdoc Warinner, where we compared the dental calculus across different primates and also humans and Neanderthals, and we're able to make inferences about the diets of early humans based on specific adaptations that those bacteria had to consuming certain nutrients. You can also learn from the enamel. So setting aside the genetic analyses, there's many other things you can do with the tooth. So we know that the way in which a tooth forms, it forms from the tip of the crown and grows to the root. And so you can do serial sampling of the enamel, and you can use a variety of different types of analyses like stable isotopes, or you can look for things like mobility through time. And you can compare that across the dentition or within a tooth. You can also, one important type of analysis that's often applied to animals is you can do cementum analysis. This can give you a sense of, you look at what stage the cementum was forming, because it has an annual formation cycle, and you can determine the season of death, for example, or season of slaughter for a particular animal. And that can help you reconstruct animal management in the past, which can be very important, for example, in the transition towards pastoralism or agriculture and management of animals within different human systems. We can also learn a lot about disease, not only from the pulp chamber of teeth, but also by looking at morphological changes that occur in the progression of a disease. For example, in cases of leprosy or tuberculosis, we can also take samples of DNA from these lesions to try to understand the causes of disease in the past. Another really exciting source of information about the past comes from paleofeces, which those of you who know my work know this is something I'm very interested in. In case you've never seen ancient poop, this is what it looks like. It's very fibrous. It contains lots of seeds, for example, and other botanical remains. You can use paleoethnobotany and microscopy to examine these and to start to reconstruct some of the foods that were available to ancient people. You can also do genetic analysis and reconstruct the microbiota to make inferences about how the gut microbiome might have changed. You can also detect plant DNA or animal DNA here as well. So here you see my postdoc Alex and student Maxime, who've been doing a lot of work on the reconstructing the gut microbiome from paleofeces. There's also a wide variety of analyses we can do with different artifacts. So I've done a number of projects in Nepal, and there we're looking at these amazingly well-preserved funerary offerings. This is a bowl, for example. It actually has preserved rice husks that you can see. The preservation is extraordinary. But we can also look at the non-identifiable residues within these vessels to try to understand what they may have contained, and to look at the origins of certain traditions, certain food technologies. So there's lots of different ways that we can approach the past. Now, until very recently, however, none of this was really visible to us. It's really only in the last decade, with a wide variety of different new technologies, that this ancient past has become seeable, has become knowable through archaeological science techniques. This, for example, is a magnification of that dental calculus I mentioned before, where we've stained the DNA using a fluorescent dye. And you can really see we've gone from not being able to access that at all to suddenly seeing millions of genomes and being able to access them just within a decade. New technologies, as I mentioned, were critical to this development. So here you see an Illumina chip, for example, which is revolutionary in genetics, but across the board in proteomics and stable isotope analysis, it's the availability of new instruments and new techniques, which has really made this truly exciting and a truly special time to be doing the work that we do. Also, massive gains in computation, just thinking about how much computers have changed over the past decade. So much of what we do today, whether that's linguistics or genetics, would not have been possible a decade ago. We simply didn't have the computational power. And we also have improved laboratories, really state-of-the-art abilities today that simply didn't exist even a decade ago. So as students, you're joining this absolutely growing, incredible, but very new field, and it's very exciting. So now I just want to return to archaeology, and I want to talk today about two case studies. I'm not going to talk about some of my main work, because I know that was kind of discussed earlier in the summer course by some of my students and postdocs who presented. And so a lot of my work on the microbiome has already been talked about, so I'm not going to talk about that now. Instead, I'm going to talk about two projects I was involved in that were particularly multidisciplinary. And I want to talk about how I think bringing in those different perspectives made such an enormous difference in really being able to understand an aspect of the past that had been previously elusive. But by bringing together these techniques, we were able to really learn something pretty extraordinary. The first is a study I'm going to talk about, sort of an accidental discovery we made about the role of women in early medieval book production. And then I'm going to talk also about what we've learned recently about the epidemics that affected Mexico during the 16th century. And then last, I'm going to talk about, and I think this is really important, that we do our research, but we also communicate our research. I'm going to talk about the importance of public outreach and communication within our field. So first, I'm going to talk about who made the books of the Middle Ages. This was a study that was only possible by putting together a lot of people with very diverse skills, ranging from archaeologists to physicists to historians. We put this team together after we made a number of unexpected findings, and then tried to put together the best team we could to try to sort out this question. And it built on itself and went in directions that were totally surprising and very exciting for us. So the way this project started it began with a study actually of periodontal disease. I was engaged in a study looking at a medieval cemetery, and we were trying to understand to what degree the causes of periodontal disease had remained similar through time. And so we've done a large-scale genetic study of dental calculus that you see here from one of those earlier photographs. But in the course of this study, we noticed something that we didn't publish on immediately because we didn't know what it was. In analyzing one of the individuals from this study, when we actually looked more closely at the dental calculus, Anita Radini, who was a PhD student on the project, noticed that there were blue flecks, these blue crystals were embedded within this calculus matrix. And this is something she had never seen before, I had never seen before. We didn't know what this was. But it immediately raised the question of, was this a pigment? Because blue minerals are very rare. It's not something you can pick up from soil. Blue minerals have to be mined. It's an uncommon type of mineral. And this raised the question of, could this be evidence of painting, of craft production? But what do we know about craft production during the medieval period? This woman that we were studying, she came from a kind of monastic cemetery, an early cemetery related to a group of religious women. We know about book production. There's lots of evidence or depictions of book production in the Middle Ages. Overwhelmingly, men are depicted as the ones producing books. And yet here we had what appeared to be a pigment in the dental calculus of this woman at this site. And so we started looking into this a lot more. And we know that women did write at this time. They were literate and they did occasionally write and paint. But so much of what we know about women's role has been affected by the way history has been passed down. Women's religious communities were much lower funded than men's. They tended to have a higher failure rate in that they didn't have as many funds over time to keep going. So a lot of their historical records were destroyed and didn't survive to the present day. Also, particularly for women, there was an element of humility where you didn't sign your work. So much of the books of the Middle Ages are unsigned, and perhaps many of those are by women, but we do not know. And there'd been a number of different types of work trying to see if women were involved, mainly using, for example, handwriting comparisons. So if you had a signed work, then trying to find that person, going through by doing handwriting comparisons. Been a lot of different attempts to try to find women artists during this period. And there had been some really fun ones. So here's an example of writing that we know was done by a woman. And I don't know if any of you can see how we know that. I'll give you just a second. If you guys can spot where on this medieval text we have confirmation that a woman wrote this text. It's true, signing was not common overall, but men signed it more than women did. So you do find signed works. All right, so here in this particular example, if you flip it around, we see that it says, Ego soror Margareta. I am Sister Margarita. This is actually a practice page where this woman was learning how to write and form her letters in a consistent way in preparation for being able to copy books. And she actually signed here at the bottom upside down. So we do have these occasional examples. We also have a few examples. So here, for example, this is the Beatus Apocalypse of Gerona. This is an illuminated page, and the manuscript. It says at the end that the manuscript was illustrated by the priests Emeterius and Enda. She's only given a single name, who was a woman and a servant of God. So this is the earliest example we know of women contributing to a book in the medieval period. There's also, for example, Hildegard of Bingen is quite well known. She was this amazing religious woman who composed different works and had many books produced of her own writing. This is the example of Civius, which she wrote, and presumably women within her monastery painted and produced. There's also this fantastic example from something called the Gouda Homilar, which is dated to about 1150 to 1200. This comes from Frankfurt. And this is believed to be the first work in Europe that's a self-portrait, a signed self-portrait of a woman. And here she writes in Latin, you can see it kind of scrolling around. She says, Gouda, a sinful woman, wrote and painted this book. This is again an example of humility. So she said she's a sinful woman, so she's not prideful. But this is one of the few examples where we have really direct evidence of a woman painting or writing a book. Here's another one. This is a little bit later. This is from the Codex Gisela. She was at a Cistercian convent. She was very wealthy, and so she was able to pay for the gold and the other very expensive pigments that you see in here. This is an illuminated nativity scene, as you can see. But if you look closely, actually, you can see that under the nativity scene, you can actually see Gisela. She's here, and she is leading the other nuns in song. But again, this is one of the other few examples of we really know that this was composed and painted by a woman. But the libraries are full of books that have been copied over and over again in order to maintain them, and we don't know very much about who produced them. So in this study, as I mentioned before, was originally a study for a very different purpose. We were looking at the site of Dahlheim, and it's a old site where there had been a church. There had been a women's religious community that had lived there. They'd been there for several centuries, but eventually the community was abandoned after the plague, and then a series of wars where it was burned to the ground multiple times, and eventually the women left it permanently. It was later bought by a men's religious community and turned into a monastery. And that monastery in the 19th century was then secularized, and it's now a museum. So it's a major museum of monasteries in Germany that you can visit today. And actually during the construction of this building here, which is a modern building, they uncovered the cemetery associated with this earliest church, the women's community that was there. And so we were analyzing the individuals that had been excavated during this construction project to try to understand the history of periodontal disease. And so we published this study, and you'll notice that the remains from this collection, they're in banana boxes. You're not seeing things, they are banana boxes. They were not previously used for bananas, but it turns out that many of these types of boxes that are utilized in, for example, in the produce production, they put pesticides in them. And so they're actually excellent for storing remains because they prevent pest overgrowth. So when we looked at this one individual, as I mentioned before, when the dental calculus was decalcified, in this case, this was an attempt to look at food remains, these blue pigments just kept falling out of the calculus, hundreds and hundreds of them. And we wanted to know what they were. Now, at the time, there were 6 different blue mineral pigments known: azurite, lazurite, vivianite, malachite, Egyptian blue, and smalt. And so we wanted to see if we could figure out which one of these they were. We assumed it would be azurite, which is a very common mineral. It's blue, and it's found throughout Europe. There's many mines that produce azurite. It's a kind of copper oxide. So we used a combination of SEM-EDS, which allows you to get elemental analysis, and also Raman spectroscopy, which can tell you something about the mineral structure in order to identify this blue pigment. Now, one thing that was really important, Anita also realized, this is a great example of being conscientious. When Anita was first doing this, the typical way that you release microfossils from dental calculus is to use an acid digestion, so to use hydrochloric acid. Most people would set it up, walk away, come back the next day after the decalcification is finished, and then look at the results. But fortunately, Anita didn't do that. She actually watched the decalcification process. And it was during this process that she would see the blue pigments emerge and then fade to gray and then turn transparent. And we didn't know what was happening. But what was going on was the acid was breaking down the blue color in these minerals. In fact, we would have never have known the blue pigment was there had she not been so conscientious in watching this. She did a number of experiments. She actually developed an alternative method for extracting these pigments that didn't break down the color. And you can see the color differences here between the hydrochloric acid extraction and the new water and sonication extraction method that she developed, where the blue color was retained. By doing that, we were able to produce enough of these, or recover enough of these blue minerals to actually then be able to analyze their composition. And so you can see here, she did a number of experiments, extracting them, and then we analyzed them. So this was the SEM EDS profile, the elemental composition that we obtained from the blue pigment within the calculus. And we then compared them to reference standards from other blue pigments. And what we see is that it's a match for lazurite. And this is really exciting because this is the blue color that makes lapis lazuli blue, which is the source of ultramarine pigment in the Middle Ages. We also were able to use Raman spectroscopy and confirm that it matched, for example, has the same lazurite spectrum, Raman spectrum as, for example, in Michelangelo's painting that you see here, and it matched reference lazurite. And importantly, some of the colorless minerals that were also there, we analyzed those because, of course, lapis lazuli isn't just lazurite. It's actually a composite mineral with others. Phlogopite is another one that's very important, and it's that association between lazurite and phlogopite that is very specific to lapis lazuli. And we were able to show that this colorless particles within the calculus as well match phlogopite. So here we had both lazurite and phlogopite. That really gives it allows us to conclusively match this to lapis lazuli. Now that raises the question, what on earth is lapis lazuli doing here? Because the pigment that you make from lapis lazuli, which is ultramarine, is an amazingly expensive pigment that originates from just a couple of mines that were known at this time in Afghanistan. So it meant that this pigment had traveled over trade routes from Afghanistan all the way to remote rural Germany to this women's religious community, all within the early, kind of 11th century, 12th century CE. And so through this, we can start to understand this really unexpected source, these ancient trade routes or these medieval trade routes that had been established for which we don't have very good records. And we certainly don't know how far these trade goods were going within rural Central Europe. And so by looking at this individual and reconstructing the presence of the lapis lazuli here, and then looking also at radiocarbon dates for her. So she dates to roughly about 1,000 to 1,200, somewhere within that range. You can see there's a wiggle in the curve, which makes it hard to be precise. We can put her in perspective of the known other women, the handful that we know who were producing books of this period, and she falls right within this. So she seems to be after Enda, but maybe a little bit before Hildegard of Bingen and Gouda and Gisela. So here we are. So up until now, we have a few examples of these what appear to be extraordinary women who may be very different from everyone else and being very distinctive in producing these books. But the finding of this woman at Dahlheim makes us think that maybe there were many, many other women who would have never been found otherwise. They didn't sign their books, maybe their books didn't survive, but perhaps this is a more widespread activity. And so this raises the possibility of going back and looking at more medieval cemeteries to try to find more artists within them, both at women's and men's communities. And so from this kind of unexpected source, we were able to reconstruct the practices and behaviors of women in medieval society, and also trace trade routes all the way back to Afghanistan. So that's example one. We're pulling together the physicists to be able to help us with, for example, the Raman spectroscopy, and the medieval historians who were able to help us put this in context. Together with the archaeologists, we were able to tell this much broader story. Next, I'm going to talk about colonial epidemics. This is an area that's really important to me. This is what I did my PhD work on initially, actually, was trying to understand the impact of European colonialism on indigenous peoples in the Americas. So we know that from 1492 through the, say, 1520, hundreds of ships came to the Americas. Columbus initially landed in the Bahamas, but then literally hundreds of ships came. And with them, they brought tens of thousands of Europeans who brought with them many diseases. We know that there were many, many epidemics that broke out within Mesoamerica and the Caribbean islands within just the first 100 years of contact even. And so here you see some of the known dates of major epidemics in the region. And we have a lot of testimony to it as well. So Bartolome de las Casas, who was a Dominican friar at the time, he recorded many of these outbreaks. For example, of the early ones in the late 1490s, he said, there did not remain the multitudes of people that were on this island from the year 1494 until that of 1496. It is believed the third of all of them. So he's reporting how many people were dying of these brand new epidemics that were being brought, to which people had never before been exposed. He writes in 1520, a terrible plague came and almost everyone died and very few remained alive. This was smallpox, which was given to the miserable Indians by some person from Castile. I do not believe that 1,000 souls escaped this misery from the immensity of people that lived on this island, and which we have seen with our own eyes. And this played a huge role. These diseases played a massive role in the conquest of Mexico, because this 1520 epidemic ripped through Mesoamerica right during the period during which Cortez was trying to conquer Tenochtitlan. And we know that it played a major role and a decisive role in the outcome of their battles. Conquistador Bernal Diaz del Castillo, for example, wrote in his famous recollections of the war, the whole country was stricken and filled with it, meaning smallpox, from which there was great mortality. We have also recordings of people experiencing this 1520 epidemic. So for example, this is from the Annals of the Caciquels. The Caciquel are a Maya people that live down here in highland Guatemala. And they record that it happened during the 25th year, meaning 1520. The plague began. First, they became ill of a cough. They suffered from nosebleeds and illness of the bladder. It was truly terrible, and the number of dead there were in that period. And the prince Vakaki Amak died. Then little by little, heavy shadows and black night enveloped our fathers and grandfathers and us also, oh my sons. When the plague raged, it was in truth terrible. The number of dead among the people could not in any way control the sickness. And great was the stench of the dead. The mortality was terrible. Your grandfathers died, and with them died the son of the king and his brother and kinsman. So it was that we became orphans, oh my son. And so what you see here is a recording not only of the disease, but the social impact. It had devastating effects, not only through population decline, but in completely collapsing the government. You have entire royal families dying from the disease. So it caused absolute political turmoil. The Franciscan friar Bernardino Sahagún also recorded many of these different epidemics. He's often credited as being the first anthropologist. He wrote a major encyclopedia of life among the Aztecs in the 16th century. And he worked with many indigenous people to record their histories. And he wrote also about the 1520 smallpox epidemic. He talked about how some were quite covered with the pustules on all parts, their faces, their heads, their breasts, etc. There was great havoc. Very many died of it, and they could not walk. They only lay in their resting places in beds. They could not move. They could not stir. They could not change positions, nor lie on one side, nor face down, nor on their backs. And if they stirred, they would cry out. Great was its destruction. Covered, mantled with pustules, very many people died of them, and very many people starved. There was death from hunger, for none could take care of the sick and nothing could be done for them. Now, this was a smallpox outbreak, and we know that smallpox has a mortality rate of about 30%. But I think this last part here is so critical, that he records that the mortality was even higher, but because so many people became ill at once, there was no one to care for the sick, to bring them water or bring them food. So many people died of thirst and exposure in addition to this. This was different in Europe, where smallpox was mainly a childhood disease, because it came through so frequently that any adults who survived would be immune because they would have already had it in childhood. So this wasn't something that in Europe caused societal collapse, because it affected only a subset of the community. Here, it affected everyone at once, and you no longer had adults that could care for their children, let alone themselves. So this also contributed to the massive mortality rates that were seen during this period. So what were some of the things that contributed to the devastation of these epidemics? Well, first of all, you had large migrations of sick colonists coming regularly. You had a complete lack of previous exposure, and therefore no immunity amongst the people who were now being exposed to these diseases. You had also a colonial policy of congregación, which was an attempt by the Spanish to gain more political control by forcibly relocating people into denser settlements that were easier to control, but also spread disease faster. You had massive social disruption from the mortality and the morbidity associated with these diseases. You had a new colonial structure put in place so that people were being forced to work, to be overworked, and were abused, which weakened their immune systems even further. all of this contributed to food shortages, and you had insufficient medical care. And also something that isn't thought about very much, but one of the diseases we know came is mumps, which although doesn't have a high mortality rate, has a high rate of causing sterility in males. And so all of this further collapsed the society. We know that 3 of these epidemics were particularly devastating. The first one that I mentioned in 1520, which had a decisive impact on the outcome of the war between the Spanish and the Aztecs, but also again in the 1550s, sorry, this one's a little bit off, should be the 1545 here that you see in central Mexico, and it's a little bit later in Guatemala. And this is also, sorry, this is moved. This should be highlighting 1576 to 1580. This was also another major epidemic. We know the first one was smallpox, and we know the next 2 were named Cocolitzli, but Cocolitzli is an Aztec word that means pestilence. It's not a specific name. And it was not clear what was the cause of Cocolitzli. And in fact, for many of these epidemics that you see here, we don't know what their cause was. We do know smallpox because the Spanish were familiar with smallpox and they named it smallpox. But the others are very hard to diagnose and are often contested. Some of the diseases that have been proposed are measles, mumps, influenza, malaria, dysentery, typhus, typhoid fever. Others, it's very unclear. Now, there is a misconception that we know, a lot of people think we know nothing about this period from the indigenous perspective. And it is true that the Spanish did destroy many, many indigenous documents. Diego de Landa famously destroyed hundreds or thousands of indigenous documents. But there are some that do survive. And so we can also see the impact of these diseases through the ideas, through the lens of indigenous peoples. So for example, this is the Codex Gisela, and here they record many of the diseases that affected them. So here, for example, you see this person with their eyes closed, that indicates death, and they're covered with a rash. And it says this year, 1537 to 1538, many people died of smallpox. You also see here for the year 1544 and 1545, it says there was a great mortality among the Indians. This is one of these, this is a depiction of stacked bodies from that 1545 Cocolitzli epidemic that I mentioned. This is the Codex Mexicanus, and here you can see the arrival of the Spanish and the conquest, and you can see 1520, that's the smallpox epidemic. You see La Malinche doing translations for Cortez there. You see the 1548 epidemic, the 1579, 1518 epidemic, this one labeled again Cocolitzli. You see here the Codex Aubin. We have other examples here. Again, that 1545 epidemic is recorded with face hemorrhaging. You see also here in 1576, the Cocolitzli is mentioned again. This is another document. This is the Codex Azcatitlan. I'm sorry. And in that, you also see these diseases depicted. So there's many hypotheses about what some of these particularly devastating epidemics may have been. And specifically, I'm going to talk now about Cocolitzli, these 1545 and 1576 outbreaks of Cocolitzli. Previously, based on medical historians, based on texts and the symptoms, had proposed that perhaps it's typhoid fever, which is caused by Salmonella enterica typhi, or epidemic typhus, perhaps plague or influenza, or some sort of unknown hemorrhagic fever. But these are all very different diseases, and both bacterial and viral. How could we possibly narrow this down? So in 2006, I had the great opportunity to go and work at a site called Tapascaloola in Mexico. This is a really interesting site. It's a colonial site. It's a site that was a major Mixtec city that then the Spanish set up a mission at in the early 16th century. But what was really unusual about this site is this is part of the administrative core. This is one of the main administrative buildings, and you can see the original floor. So we were excavating down to the original floor and trying to find the basic kind of political structure, the architecture of the political center of this colonial city. But what we found is when we pulled back this overburden of vegetation, the original plaster floor had all of these cuts in it, these kind of long oval cuts. And when we excavated down, we realized that these were each mass graves. In fact, this entire administrative center was full of mass graves. And we actually know a lot about this site. In fact, this is a drawing here of the local lord and his son of Tapascaloola. We know their names. We have quite a few records from this site. There was a major trial, an Inquisition trial actually took place here. So it's very well documented in many ways. But what was unexpected was to see these graves. What we now know is it is the first and only confirmed epidemic cemetery that has been excavated in Mexico, and we can date it to the 1545 Colonial epidemic. We did a number of test excavations to see where we have, to make sure that kind of how full this plaza was full of graves. It does appear that everywhere we excavated, we found mass graves, and so we think the entire plaza was a cemetery. Each grave contains between 1 and 6 individuals. And so they were probably dug daily during the course of the epidemic. This is a drawing of one of the individuals that we found. This particular grave had 3 individuals in it. And we wanted to know, could we identify from these remains the cause of this epidemic? Now, doing this is like finding a needle in a haystack. It's really difficult. How are you going to find the pathogen DNA amongst everything else when you don't even know what the pathogen is? And so there had been many attempts to do this. And actually, after I finished my PhD, I hadn't been able to solve it. And later, other groups tried to solve it and tried to understand what type of pathogen it was. And they also failed. The technology, this is in 2014, just wasn't good enough to be able to identify it. One thing that was really exciting, though, is here at the MPI, Johannes Krause and Alexander Herbig and Kirsty Voss were developing new technologies that were much more sensitive and using different approaches that offered the first hope of really being able to understand what the cause of this epidemic had been. And so this is work I'm going to show now led by Ash Vaganay, who's now in Copenhagen. And it was really because of the advent of next generation technologies, plus also new computational tools, malt and hops. Hops was developed actually during this study and was published a little later, that we were able to, and combine that with capture enrichment techniques, we were able to actually be able to identify the pathogen, which you see here in this plot. So everything you see here in red is Salmonella enterica. But what was really interesting is instead of being Salmonella enterica typhi, typhoid fever, which has been previously proposed, what we actually found was that it was a type of paratyphi C. This is a variant of Salmonella enterica that today doesn't cause epidemic disease. In fact, it's almost completely gone. It was nearly eradicated with the advent of antibiotics in the early 20th century. And as a result, we know very little about it. It was not on anyone's radar, and we would have never expected that to have been the major cause of disease in the Americas. It's closely related to other types of Salmonella enterica that infect pigs. And so it's possible that it was a zoonotic or related to the pigs that were brought with the Spanish. It was also then found within Europe, and we now know it was endemic in Europe at that time. And so this might have been something that was circulating between humans and animals and then was introduced during the colonial period and then spread to epidemic proportions. Now, I'm really interested in carrying this work forward because we have, as I mentioned, there is a lot of documentation of the 16th century in Mexico, including of disease. And there's a particular set of documents called the Relaciones Geográficas, which provide kind of city or town by town level information about disease that occurred in Mesoamerica. And so I've been working with a group of undergraduates at Harvard and Harvard Medical School to try to look for these historical references to different types of illness and disease. So far, we found references to smallpox, kokolitzli, dysentery, lots of fevers, malaria, syphilis, and also things like mental health, like depression and suicide. And now what we're working with Rodrigo Barquera on is trying to now combine this information. Can we take the historical information and then go into the archaeological record and try to look for these specific diseases and link them to specific outbreaks? All right, now last what I want to do is just talk a little bit about the importance of collaboration and communication and outreach. So I think it's so important because this field is developing so quickly, so fast, and it's so exciting, but sometimes it goes too fast. We aren't really effectively communicating across disciplines, and that is so critical to help other people across different fields of study understand what you're doing, both to create better future collaborations, but also to help explain what we're doing. So Courtney Hoffman, who's a former postdoc of mine. She's now a professor at the University of Oklahoma. We've been doing these Ancient DNA 101 seminars through the Society for American Archaeology. We're just trying to explain what some of these new developments are that are happening at such a rapid pace in the field, so that archaeologists know what's possible, because it's constantly changing. Things we said were impossible 3 years ago are possible today. So trying to get the word out about how the field is changing and what is now might be possible is really important. And it's also important too, because it's very easy for miscommunications to happen across the field, and that can happen on both sides. Sometimes in archaeological science, we might take a naive view to archaeology or prehistory. So it's so important that we work together and we work across disciplines. And so it's through these types of communications at conferences and other opportunities through outreach projects that becomes critical to holding our field together as we grow so quickly. There's a lot of really exciting opportunities that we can do too that are just now being explored. And this is something I'm really excited about. You know, we do work all around the world, and the standard language of publication for scientific publishing is English. But of course, we work on topics that are of interest to people in many other places. And also we're doing work on the heritage of countries around the world. It's really important that those stories also be available to the people that we're working with and in their own language. So we've been on some of our projects, we've been working with our collaborators and others to translate the abstracts into the languages that are spoken in the place where we are doing work, and in some cases, whole articles. We've also been experimenting with producing artistic reproductions to show what we're seeing, to show aspects of kind of reimagine ancient cultures. This is an example that we had produced for the Xiongnu, which was a major study that we did, which is a population that it's an ancient society that lived between about 200 BC and to 200 CE in Mongolia. And we had a number of findings that were exciting and really new about the Xiongnu. And so we wanted to depict this through art, really give them a kind of visual representation. The Xiongnu were well known in China as being these nomadic warriors. And so much of the historical recording of them is quite negative, because they were raiding. And they're often depicted as being very warlike. We wanted to use our findings, which were much more about the mundane, the everyday, the cooking, to show them in a way that wasn't solely through the lens of war. We can also experiment with different forms of press releases or local press releases. This was a great idea by my student James, and also worked on by Irina. We did a project where we worked with many, many different countries to reconstruct the evolutionary history of our oral bacteria. And we had this translated into a summary of the paper into all the languages of the different countries that participated in our study. And I thought this was a really great idea and really exciting. And then last, my students and I started this coloring book project, which has been just really fun. And I think a great example of how we can give back to different communities and also explain what we do. So we produced this coloring books. This is entirely produced by students and postdocs at the Max Planck. And we now have partnered with different archaeologists and linguists and biologists around the world to translate it into more than 30 languages. So here you see Arabic, Tibetan, and I believe this is Yucatec Mayan. And trying to get this into as many languages as possible so that people around the world can learn about the work that we're doing in their own language. And then last, I just want to highlight some of the really great work that's being done by students. So this is Zandra Fagerness, one of my students who's doing a soapbox science presentation about what you can learn from dental plaque. And then these are a couple of videos made by Rodrigo Barquera showing how ancient DNA analysis works in the lab and translated into Spanish and Nahuatl. Again, we have the great privilege of being able to work on these amazing stories from history. And I think we have a responsibility to then communicate what it is we're actually doing and what it means, and to do it in the languages that really reach the people that most matter for these studies. So with that, I will close, and I'd be happy to take any questions, but I'm just I want to say thank you so much to the organizers of this summer school. I think it is such a fantastic idea, and I know it was a lot of work to pull off during the pandemic, but thank you so much for having me, and thank you so much for organizing this.